Hello, my name is Andrey Prjibelski and I am from St. Petersburg State University. The title of my talk is IsoQuant, Isoform Analysis and Quantification with Long Error-Prone Transcriptomic Reads. I will talk about our tool that we designed for long read RNA analysis. The pipeline is pretty simple. We take as input a gene annotation and long read alignments that you can obtain with your favorite spliced aligner, for example Minimap, Gmap or StarLong. Then we match align reads against known transcripts. We try to assign each read to a known isoform, detect inconsistencies between mapped reads and known isoforms, and count different features such as genes, transcripts, and exons. Further, we split reads into two different categories: consistent splice matches, which most likely represent known transcripts, and inconsistent splice matches, which most likely correspond to novel transcripts or events such as intron retention. We further group these reads by their inconsistency type and attempt to create novel transcript models. Unfortunately, due to the lack of time, I won't go deeper into algorithmic details. However, if you are interested, you can always check our GitHub link at the end of the presentation. We tested IsoQuant on both real and simulated data. Simulated data was generated using two different tools: IsoQuant, which is good for simulating PacBio data. It inserts errors into reference sequence randomly and uniformly. TransNanoSim is a bit more sophisticated. It learns on real data and then attempts to introduce platform-specific sequencing errors in artifacts typical for nanopore sequencing. We tested IsoQuant on reference transcripts, which is a perfect case. We also generated PacBio data using IsoQuant with error rate at about 1.5%, and we simulated nanopore data with error rate at about 11%. Nanopore data was simulated using both IsoQuant with uniform error distribution and with TransNanoSim with platform-specific errors. So, of course, TransNanoSim simulated data is more realistic and closer to the real nanopore data. Error rates were derived from real PacBio CCS and nanopore cDNA sequencing data. Firstly, we compared IsoQuant to Scanty 2, which does pretty much the same job, assigns known reads to known isoforms. If you take a look at reference transcripts or PacBio data, you see that both tools have very high precision and decent recall. However, if you take a look at nanopore data, you see that precision of Scanty drops. The thing I want to point out here is that mouse data was generated with TransNanoSim, so it seems to be more realistic comparing to real data. However, human ONT data was simulated using IsoQuant with uniform error distribution. And although they have the same error rate, the precision for mouse data is significantly lower. And the same you can say about the recall on mouse data, which means that the type of errors you have in nanopore data is more important than the overall error rate, and those sequencing artifacts impact the results significantly. However, positive thing about IsoQuant is even on more complex nanopore data, it manages to keep precision at a very high level. The second experiment we wanted to carry out is to assess how well we construct transcript models from long reads. For this purpose, we simulated long reads in a similar manner from the entire gene annotation, but also created a partial annotation by removing about 5% of spliced transcripts randomly. So we have long reads for the entire transcriptome and incomplete gene annotation. And we feed both into the tool that is capable of constructing transcript models. And then we compare their results with GFF compare to estimate sensitivity and precision. In this experiment, we compared IsoQuant with StringTie2 and Flare. We are also aware of a reference-based assembler called Scarop ALR, which is capable of constructing transcript models from long reads as well, but it requires PacBio CCS header file as an input, so it's not easy to run it on simulated data. As you see here, on both PacBio and nanopore data simulated with IsoQuant with uniform error distribution, all tools generate decent precision and sensitivity, with IsoQuant having a fraction higher precision than flare and string tie. However, again, coming to more realistic nanopore data, you see that precision and sensitivity drops significantly, but IsoQuant manages to keep precision at a very high level, higher than 99%, similar to PacBio. In addition, we wanted to see how consistent the results of these three tools, and it appeared that they are not very well consistent. Only about 10% of generated transcript models is common for all three tools. The positive thing about IsoQuant is that we generate no transcripts that are not supported by any other tool. These results were obtained using GFF compare as well. And of course, you can test your tool on simulated data as much as you want, but you always have to check it on real data as well. So we decided to use human, publicly available PacBio CCS and nanopore direct RNA sequencing data. As expected, the percentage of uniquely assigned reads is higher for PacBio CCS, I would say significantly higher, and the number of reads that were matched with inconsistencies is higher for nanopore data. And of course, it affects the number of novel transcripts discovered for nanopore data. Similarly, as for simulated data, we decided to see whether the results of flare, string tie, and IsoQuant are consistent on real data, and it appears that they are not again. For PacBio data, we have only about 8% of common transcript models, and for nanopore, the number is about 7% of transcript models constructed by all three tools simultaneously. And again, the positive thing that we like about IsoQuant that it does not create a lot of transcript models which are not supported by other tools, about 0.1% for PacBio and 3.7% for nanopore. And of course, on real data, you don't know the ground truth, so we cannot say which tool is correct and which is not here. So that's it for today. As a take-home message, I would like you to hear that we created a new tool for long-read RNA analysis, which shows high precision on both PacBio and nanopore reads. We still think that analyzing nanopore RNA data is a big and open challenge. And we also see that tools that generate transcript models from long reads are not very well consistent with each other. Thank you very much for your attention. If you are interested, check out our GitHub link, and thank you very much.